This will be a walkthrough of the AQA GCSE Combined Science Biology 2023 Higher Tier Paper 2. Question 1. Sexual reproduction in humans involves the production of egg cells and sperm cells. 1.1. Name the type of cell division that produces egg cells and sperm cells. So we're dealing with cell division. So what we have learned are two types of cell division, which is mitosis and meiosis. But they're producing egg cells and sperm cells. Now, what do these two cells have in common? Well, they're both specialized cells, but they both contain 23 chromosomes. Now, that is half of what a body cell would have, because a body cell would have 46 chromosomes. So what would need to happen is the egg cells and the sperm cells would need to join together, because 23 times 2 would give us 46. So we need to remember then which type of cell division will produce cells that have half of the normal set of chromosomes that a body cell would have. Well, if we look at mitosis, mitosis is therefore growth and repair. So if we're trying to repair part of our body, we would expect the chromosomes that are in those repaired cells to have 46 chromosomes. So it can't be mitosis then, so it must be meiosis. Question 1.2. Sexual reproduction produces offspring that are genetically different from each other. Give 2 reasons why sexual reproduction causes variation in the offspring. Well, the first key term here is sexual reproduction. You need to remember what that is. It is the fusion of gametes. Now, what are our gametes in this case? Our gametes will be our sperm and egg cells. So as I said before, the egg cell and sperm cells need to fuse together, and that will give us organisms or cells that have 46 chromosomes. But they are genetically different from each other. The offspring are. Now, the reason why the offspring are genetically different from each other is because these sperm and egg cells that are formed are going to be different. And what do we mean by different? We mean that their DNA is going to be different, and that's why it says genetically different. So our first point needs to be that the gametes, which are egg and sperm cells, are going to be genetically different. Now, what would be a second reason why there's variation in the offspring? Now, we've defined what sexual reproduction is. It's the fusion of gametes. But in this case, then, what is actually fusing is what the egg cells and sperm cells contain. So, it's the chromosomes or the DNA, which is what the chromosomes are made from. So when these gametes fuse, their DNA is actually going to combine or mix. So when they mix, that creates different offspring, and that's what variation is. So our second point is going to be that there is mixing of DNA. Polydactyly is an inherited disorder. The allele for polydactyly is dominant, capital D. A person with 2 copies of the allele, lowercase d, will not have polydactyly. A person with the genotype capital D is homozygous. What word describes the genotype capital D, lowercase d? So let's look at this sentence above. A person with the genotype capital D is homozygous. Homozygous means that the alleles in the pair are the same. So the term homo means same. Now if we look at our genotype in our question here, the allele, capital D and lowercase d, are different. So we need to remind ourselves now what term means that the alleles in the pair are different. Well, if homo is same, then different is hetero. Well, we just add on the ending part, just like homozygous, so it's going to be heterozygous. Question 1.4. A person with the genotype capital D lowercase d and a person with the genotype lowercase d plan to have a child. Determine the probability that the child will have polydactyly. You should complete the Punnett square diagram. Identify any offspring genotype that would have polydactyly. 5 marks. So as it says, we need to complete this Punnett square right now. So what we're going to do is we're going to put the first genotype at the top here and the second genotype on the side here. So the first genotype that we're using is capital D lowercase d. But when we're looking at the parents now, because these two genotypes are from the parents, we need to separate them in the boxes. So separating the genotype for the first parent is going to be capital D in this box and lowercase d in this box. We're going to do the same thing, but for the other parent now. So it'll be lowercase d in this box, and again lowercase d in the second box. What we're then going to do is we're going to find the genotypes of the offspring. So what will be the genotype, the pair of alleles that will exist from these two alleles coming together? Well, it will be capital D, lowercase d. Then for these two alleles, I'll be lowercase d, lowercase d. And for these two alleles, it will be capital D, lowercase d. And then a final combination, and then the same. So it will be lowercase d, lowercase d. So we've completed the Punnett square diagram now. We just need to do the second bullet point. Identify any offspring genotype that would have polydactyly. So going back to the information at the top of the question, it says the allele for polydactyly is dominant, capital D. So that means that we're looking for any offspring that have one capital D in their genotype. So looking at all the offspring here, we can see that there are 2 offspring that have this capital D. So what we will do is we will circle those 2 offspring. There is one and there is another. So 2 of the offspring have polydactyly. How many offspring in total do we get in a Punnett square though? We get 4. So 2 out of the 4 have polydactyly. So that can be our probability. What will be better is if we simplify this, so divide both sides by 2, so it'll actually be a half. But you could write that as a percentage, and a percentage would be 50%. Or you could write that as a decimal, and that would be 0.5. So any of these are acceptable ways of writing a probability. 1.5. Embryos can be screened for the alleles that cause inherited disorders. Give 2 advantages of embryo screening. So, the word screen. Screening in the case of embryos really means to check. So, embryos can be checked for the alleles that cause inherited disorders. So that would be an advantage. That should be our first one that we can write. Check whether an embryo has a disorder. Now, what is the advantage of checking whether an embryo has a disorder? What can that allow you to do? Well, this embryo is going to come from 2 parents. And ultimately, the parents want to have a child. Now, by them checking whether their embryo has a disorder, it will then mean that they can prepare for life with a child that has an inherited disorder, because there may be something that they will need to do to ensure that their child is A, taken care of properly, and B, having access to things that allow them to live an easier life. So the second advantage is parents can prepare for life with a child that has a disorder. Now, another possible option is the opposite. The parents could, in this number 2 case, prepare for life with their child with a disorder, but the parents could then decide that they don't want to have a child if they're going to have this disorder. So they can go through termination of this embryo or potential life. So they have a choice of doing that. Question 2. It is estimated that 99.9% of all species that have ever existed are now extinct. 2.1. Why is the percentage of species that are extinct only an estimate? Tick one box. Well, the first option says all individuals of one species have the same genes. Well, they have the same genes, but they may have different versions of that gene. So that would actually mean alleles. But that's not going to be the reason why that's an estimate. Second thing says extinction is always caused by humans. That's definitely not true, because extinction can be caused by disease, can be caused by natural disasters, loss of habitat. So it's not only through human intervention that causes extinction. The third one says humans have not found evidence of every species. And this makes the most sense because organisms have been around for billions of years. Humans have not. So humans can only find evidence of species that have either been around during their time or have left evidence of their remains, and that's what we call fossils. But human beings have not found all fossils, so they can only make estimates based on what they see. Question 2.2. What evidence is used to study species that have become extinct? Well, as just previously mentioned, what can humans study to tell that organisms that have been extinct were actually there at one point in time, they can study the fossil record, so fossils. 2.3. A bacterium called C. difficile can infect the human digestive system. C. difficile can multiply and produce toxins. These toxins can cause diarrhea. Doctors are concerned that new strains of C. difficile may evolve. Antibiotics may not be able to kill these new strains. Explain how the evolution of antibiotic-resistant C. difficile can be slowed down. 6 marks. So let's analyze everything here again. A bacterium. So we're dealing with a certain type of microorganism. Can infect the human digestive system. C. difficile can multiply and produce toxins. So multiplying being reproduce, and toxins are the little chemicals, poisons that can damage tissues on the body. The toxins cause diarrhea, so this will be the symptom that we will experience if infected by this bacteria. Doctors are concerned that new strains of C. difficile may evolve. So evolve means change over time. Antibiotics may not be able to kill these new strains. So antibiotics are drugs that's used to kill bacteria. If they're not being able to kill these new strains, that must mean that the bacteria are resistant. So, explain how the evolution of antibiotic-resistant C. difficile can be slowed down. So, trying to reduce the rate. So, we can start by thinking about the antibiotics itself. Well, we said that it's a drug that's used to kill bacteria. But antibiotics are prescribed by doctors. Now, when someone is prescribed an antibiotic, one thing that they must do is they must follow their prescription and complete it. So, first thing that we can say is that patients that are given the antibiotic must complete their prescription. Now, we have to explain. So, we've said a point, but we haven't given the reason why that patients must complete this. And remember what the antibiotic is there to do. The antibiotic is there to kill the bacteria. If they complete the prescription of the antibiotic, it should mean that all of this C. difficile in this specific case should be killed. Now, as it said in the question, the bacteria can infect the human digestive system. Now, if there is a bad infection, then it would make sense to prescribe a patient an antibiotic. But if it's just a minor infection, then there is no need for an antibiotic. Eventually, the patient will heal and recover over time. If you prescribe an antibiotic for a mild or minor infection, then it may cause the development of these bacteria that are resistant to the antibiotic. Now, what's the reason how antibiotic-resistant bacteria can exist? It's through mutations. So, by not giving patients with minor infections this antibiotic, it reduces the chance of bacteria mutating to become antibiotic resistant. So the next thing that we can say is, do not give antibiotics for minor infections. And our reason is to prevent the chances of bacteria mutating, becoming resistant. Next point, the antibiotic is supposed to kill bacteria. That's why it's designed. But that means that it's not there to kill any other type of pathogen, for example, viruses or fungi. So, patients should not be prescribed antibiotics for viral infections. Our reason is because antibiotics do not work against viruses. And one more point that we could say is, how can we prevent the spread of this C. difficile? Well, to prevent spread of a pathogen, there are some methods that human beings can do. One of the most common methods is to wash your hands. Now remember that the toxins cause diarrhea. So a person that's experiencing diarrhea would most frequently go to the toilet. So what we should say is to wash hands thoroughly after using the toilet. And our reasoning again is to prevent the spread of C. difficile. So for each of the points that we have stated, we've given a reason, and that will allow us to answer the question properly because we've explained, so we can get 6 marks. Typically for a question like this, you want to get at least 3 points, 3 reasons, and that should satisfy 6 marks. Question 3. Figure 1 shows a river next to a field. 3.1. Describe a method to investigate how the distance from the river affects the number of different plant species in a field. You should explain how to use a transect in your method. 4 marks. So this is related to your required practical of field investigation. Now in this practical for field investigation, there are 2 methods for this. Method 1 is where you're trying to find the abundance of a particular plant species in a field. Method 2 is when you're trying to find the distribution of a particular plant species in an area. Now, if you're trying to measure the abundance, the main piece of equipment that you can use is a quadrat. You only need to use a quadrat to find the abundance. You don't need to use any other piece of equipment to do that. But when you're trying to find out the distribution of a species, you want to find out how does that plant species change in its number from one area to another. Now, for you to measure that, you need to use 2 pieces of equipment. You need to use a transect and you need to use the quadrat. So what we want to find out now is which of these 2 methods do we need to do for this example here. So let's read it again and see if we can identify it. Describe a method to investigate how the distance from the river affects the number of different plant species in the field. So we're looking to see different plant species from the river, so that means going away from the river, affects different plant species in the field. So that means that we are looking at the distribution. And the hint is by them telling us that we have to explain how to use a transect. So the first thing that we would need to say is we need to place our transect down at a certain place. Now, by looking at this arrow here, this is a perfect way that we can place down our transect. because we've placed our transect at 90 degrees or perpendicular from the river. It's going away from the river, so we know that anytime that we measure, it's going to be a certain distance away from where the river is. So our first mark will be place transect down at 90 degrees or perpendicular from the river. Now, we still need to count because it says we need to find the number of different plant species. So for us to find the number, we still would need to use our quadrat. So we're going to place the quadrat down. But remember that we have to find out how the number changes as we go from the river. So we would need to place our quadrat down and count the number of different plant species within the quadrat. But after we do that once, we then will need to move the quadrat a certain distance away from the river. So we could use that first box as the first place in which we place our quadrat, and then we can use this second box as the second place where we place our quadrat. We repeat, we count the number of different plant species in that quadrat, and then we do it again. But the interval, or the distance between where we've placed these quadrats, should be regular or even. So place quadrat down at regular intervals. And again, remember, it's from the river. Once you've placed it down, we're going to count the number of different species in the quadrat. But once we've done that, what will we do next? Well, if we think about what we've done now, we've taken a transect and we've been measuring along this transect. But remember that the river is different in terms of its length. So we've just gone in a straight line one part of the field. What would be then best would be for us to repeat this, but at a different location from the river. So we could do it there, but repeat the same thing. So take your quadrat, regular intervals, and count the number of different plant species. But the more times that you do it, the more reliable your result would be. So you would then, after we do that a second time, try to do that one more time. That would allow us to then get some more reliable results. So the last thing that you want to say is repeat with at least 2 more transects. 3.2. Students used a valid method to investigate how the distance from the river affects the number of different plant species in the field. Figure 2 shows the results. So we've got a graph. Y-axis, number of different plant species. X-axis, distance from the river in meters. We've got 6 distances, and at each distance we have the mean shown by the cross. We've got the maximum shown by the upper bar, and the minimum number of plant species shown by the lower bar. What is shown by the data in figure 2? Tick one box. Fewer different species are always recorded nearer to the river. So, but this statement means that as the number is lower, we should always expect everything to be lower. So that means our mean should always be lower, our minimum should always be lower, and our maximum should always be lower. But what we can see is that our minimum is always lower, our mean is lower, but our maximum is not. So this is not correct. The mean value students can be most certain about is 5 meters from the river. So we need to focus on 5 meters from the river. It says the mean value that students can be most certain about. Well, for the students to be most certain about the mean, for us to figure that out, it would mean that the maximum and minimum should be the closest to the mean. And the best way for us to view that would be for the bars to be shortest. And in this case, if we compare 5 to all the others, it is the shortest. So this is correct. Third one. The number of species recorded at 6 meters from the river is anomalous. So we're looking at 6 meters now. And the word anomalous means does not fit the pattern. Well, let's have a look at our pattern in terms of our mean then. We can see that there's a trend in that it's increasing. So for each distance from the river as it increases, the mean increases. So that does fit the pattern at number 6. So that's not correct. So we can definitely confirm that our second one is the right answer. Cows walk on the ground near the river more than they walk on the ground further from the river. 3.3. Which is an abiotic factor that could affect the number of different plant species found near the river? Well, for us to answer the question properly, we need to know what the keyword abiotic means. Abiotic means non-living. So we need to go through our list now and decide which one is non-living. Remember that the opposite to abiotic is biotic, which is living. So we can probably assume that some of our answers are going to be biotic factors, but we just need to identify the non-living or abiotic factor. So first one, microorganisms near the roots. Well, microorganisms could be bacteria, fungi. So this would be a living factor, biotic factor, so that's not correct. Moisture levels in the soil. Well, moisture is to do with water. So that is a abiotic factor, so that could be a correct answer there. Oxygen concentration in the air. Well, oxygen is non-living definitely, so that also is abiotic. Primary consumers in the field. Well, consumer, as primary consumer, is going to be something that is living because I would eat a producer. So that would definitely not be correct. So we're left with moisture levels in the soil and oxygen concentration in the air. So it can only be one correct answer. So let's read the question again and see if there's something in there that can help us choose the right answer. Which is an abiotic factor that could affect the number of different plant species found near the river? Well, what's going to help the plant species to grow? Because if it helps the plant species to grow, we would expect more of them to be there. Well, moisture levels in the soil, that will definitely help the plants to grow because the plants need water and they get that from the soil. Oxygen concentration in the air. Well, plants don't need the oxygen concentration to be very, very high because the plants produce the oxygen themselves. They produce the oxygen in photosynthesis, and then they use that oxygen that they make in respiration. So figure out then that it can't be the third one, so the second one would make most sense. .4 Increasing numbers of cows are being farmed across the world. Explain the environmental implications of increasing numbers of cows being farmed. 6 marks. So again, we've got a question where we need to give the reasons for something happening. But in this case, we're looking at environmental implications. So implications means consequences. And we're dealing with environment. So we could be thinking along the lines of temperature. We could be thinking land. We could be thinking air, water. These are all linked to the environment. So, key thing is cows then. Well, cows, we know that they live on land. They live on land and they're there too. And while they're on the land, they eat grass. Now, if they're eating grass, that means then that there's going to be some more plants that's going to be eaten. So right away, there's a implication of more cows being farmed. If more cows are being farmed, there's more plants that's going to be eaten. Now, what is the problem? Because remember, we're dealing with the consequences of these cows being farmed. If there are more plants eaten by cows, then there's going to be less land available. And if there's less land available, there's going to be less land available to grow crops because more of it is going to be used to farm and house these cows. Okay, next point. Cows are animals, just like humans. They're going to respire. When they respire, just like humans, they will release carbon dioxide into the atmosphere. So if there are more cows being farmed, there'll be more carbon dioxide released from them into the atmosphere. Now, not only will the cows release carbon dioxide, but they're a bit different because they will release methane into the atmosphere also. So more carbon dioxide will be released into the atmosphere and more methane will be released into the atmosphere. Now, what's the environmental implications of more carbon dioxide and methane released into the atmosphere? Well, both carbon dioxide and methane are examples of greenhouse gases. And what are greenhouse gases now? They are gases that would cause the trapping of radiation in the atmosphere. And what that will lead to, it will lead to global warming. So if there are these greater amounts of carbon dioxide and methane, that would mean that the effect of global warming will be greater. Remember that global warming is the average increase in the global temperature of the Earth. Now, what will that look like? Because remember, it's environmental applications we need to focus on. So, we've spoken about the temperature, but how will that affect land, for example? Well, if there's global warming, it will lead to destruction of habitat. For example, polar ice caps will start to melt. That could lead to flooding and loss of habitat. That could also lead to drought. So these are consequences as a direct result of global warming. Now, if there's flooding, that is also linked to what we said before. It could lead to less land available to grow crops. Let's try and include one more example. Now, if we want to house more farms so we can raise more cows, we would need to create more land. Now, for us to create more land, at this current point, most of that land is going to be occupied by trees. So what that would lead to, it would lead to deforestation. But what is the consequence of deforestation? Well, we know that one of the consequences of deforestation would be lead to an increase in carbon dioxide in the atmosphere, but we've already mentioned that. So is there anything different? Well, an increase in deforestation would lead to a decrease in biodiversity. Remember that biodiversity is a variety of different organisms, different species in an ecosystem. A reduction in biodiversity has a big impact on food chains and could also lead to a loss of different species as a result of a lack of food. So that's a major implication that affects the environment. So similar to the previous 6-mark question, we need to give our points, give our explanation for it. And I said before that you need at least 3 points with relevant explanations to try and get 6 marks. So we've done that here. We've got a point with a reason, a point with a reason, a point with a reason, a point with a reason.